Hello everyone. Today I'm going to present you a lecture on citric acid cycle. In biochemistry, a central question we ask is how the food is converted to ATP. Extraction of energy from the food occurs in 4 stages. In stage 1, nutrients are broken down to small building blocks. For example, carbohydrates are broken down to glucose. Proteins are hydrolyzed to release amino acid. Fats are hydrolyzed to release fatty acids. In stage 2, small building blocks are metabolized through various metabolic pathways and converted to a common intermediate, acetyl-CoA. Acetyl-CoA carries 2 carbons. In stage 3, citric acid cycle or TCA cycle. Break down acetyl-CoA to release 2 carbon dioxide. The energy is conserved in high-energy compound NADH and FADH2. In stage 4, these high-energy compounds are further oxidized, and the energy is used to convert ADP to ATP through electron transport chain and oxidative phosphorylation. Today, we will focus on citric acid cycle or TCA cycle. The first question we ask, why TCA cycle is important? TCA cycle is important for 2 reasons. First, most of high-energy compounds, including NADH, FADH2, are derived from TCA cycle. And only small amount is derived from various metabolic pathways in stage 2. Second, several metabolic pathways exist to break down small building blocks, but there's only one way to break down acetyl-CoA. That's through the TCA cycle. We can imagine what will happen if TCA cycle is blocked. The person will die. We have seen patients with enzymatic defects in glycolysis or beta oxidation, but it's extremely rare to see patients survive with enzymatic defects in TCA cycle. In other words, TCA cycle is essential for life. Now, let's take a closer look at the TCA cycle. The overall results of TCA cycle can be summarized in one reaction. Acetyl-CoA that carries 2 carbons is broken down to 2 carbon dioxide. Energy is conserved in high-energy compound. The detail of TCA cycle is shown here. TCA cycle is comprised of 8 reactions catalyzed by 8 enzymes. At this point, you may wonder if the whole purpose of TCA cycle is to break down acetyl-CoA. Why can't we simply break carbon-carbon bond directly in 1 or 2 reactions instead of 8? Well, in organic chemistry, mechanisms do exist to break carbon-carbon bond directly, but that requires a compound to have at least 2 functional groups. Acetyl-CoA is just too small with only 1 functional group. To solve this problem, the strategy our body adopts is to borrow a compound combined with acetyl-CoA to make it bigger, and then we break it. After the job is done, we return the compound back. This is how it works in TCA cycle. The compound we borrowed is oxaloacetate. that carries 4 carbons, combined with acetyl-CoA, 2 carbons, that give rise to citrate with 6 carbons. A minor structural rearrangement leads to isocitrate. We are ready to break first carbon-carbon bond and to release the first carbon dioxide. The result is alpha-keto glutarate with 5 carbons. We continue to break another carbon-carbon bond and to release a second carbon dioxide. The result is succinyl-CoA with 4 carbons. Removal of CoA leads to succinate with 4 carbons. The rest 3 steps is to convert the 4-carbon compound back to oxaloacetate. The key features of oxaloacetate is alpha-keto group. How can we generate an alpha-keto group within a 4-carbon compound? And this is done by following classic mechanism in organic chemistry. Oxidation generates a double bonds seen in fumarate. Hydration of the double bonds gives rise to a hydroxyl group seen in malate. Oxidation of the hydroxyl group gives rise to alpha-keto group seen in oxaloacetate. We now return the oxaloacetate back. We are ready for the next cycle. TCA cycle requires 8 steps. Are there any steps more important than the others? Of course, every step is important. As I mentioned to you earlier, enzymatic defects in TCA cycle is incompatible with life. But for U.S.M. Lee, are there any steps more important than the others? The answer is yes. It turned out 2 steps that associated with release of carbon dioxide are more important than the others. Here are the enzymes. First one, isocitrate dehydrogenase. Second one, alpha-keto glutarate dehydrogenase. These are high-yield enzymes. The rest are low-yield enzymes. Next, we ask, where does TCA cycle take place? TCA cycle takes place inside mitochondria. Most enzymes are found in the matrix of mitochondria, except one, succinate dehydrogenase. And this enzyme is located in the inner membranes of mitochondria. The overall energy yield from TCA cycle are equivalent to 12 ATP. Specifically, TCA cycle generates 3 NADH, 1 FADH2, 1 GTP. 3 NADH is equivalent to 9 ATP. 1 FADH2 is equivalent to 2 ATP. 9 plus 2 plus 1 equal to 12 ATP. Here are the steps that generate this high-energy compound. TCA cycle plays a central role in metabolism. It is connected to many metabolic pathways. Here are some examples. The first connection is TCA cycle is linked to electron transport chains. The high-energy compound generated from the TCA cycle are funneled into electron transport chains, and they are the substrate for ETC. Here there's one step particular. The step catalyzed by succinyl-CoA synthetase utilizes GDP as a substrate to synthesize GTP directly without going through oxidative phosphorylation. This type of phosphorylation is called substrate-level phosphorylation. We have seen this before in glycolysis. TCA cycle is linked to electron transport chains in another way. The enzyme succinate dehydrogenase. This enzyme is also a component of electron transport chains. Specifically, this enzyme is complex 2 of ETC. Two more examples. Succinyl-CoA can be utilized to synthesize heme. Heme is an important component for many mitochondrial enzymes, as well as for hemoglobin. On the other side, malate can be shuttled back to cytoplasm and used for gluconeogenesis. Now, let's look at these enzymes. Alpha-ketoglutarate dehydrogenase. This enzyme requires 5 coenzymes. I use 1 mnemonics to describe these 5 coenzymes. Tender loving care for Nancy. T represents thymine, L lipoic acid, C coenzyme A, F FAD, and NAD. As you may recall, another enzyme that I described to you before also requires these 5 coenzymes. And that enzyme is PDH, pyruvate dehydrogenase. This enzyme catalyzes conversion of pyruvate to acetyl-CoA. The third enzyme that also requires these 5 coenzymes is BCKDH, branch chains alpha-keto acid dehydrogenase. This enzyme catalyzes degradation of branch chains amino acids. Deficiency of any of these coenzymes would lead to inhibition of these 3 dehydrogenases. Since most high-energy compounds are derived from the TCA cycle, inhibition of alpha-ketoglutarate dehydrogenase will have most significant impact on overall energy yields. Reduced energy accounts for various symptoms caused by deficiency of these coenzymes. Here are the 2 examples. The first example, arsenic poisoning. Arsenite binds lipoic acid and blocks the 3 dehydrogenases that I described to you earlier. And arsenite also interferes with another enzyme in glycolysis. The consequence of arsenic poisoning is reduced ATP production. That can explain headache, confusion, diarrhea, drowsiness seen in acute arsenic poisoning. Another characteristic of arsenic poisoning is garlic breath. When arsenic poisoning is chronic, convulsion and missed lines may occur. These are the missed lines. Although missed lines are not specific to arsenic poisoning, they can be used as a sign for diagnosis. Second case is thiamine deficiency. It is most common in alcoholics. Thiamine is important coenzymes for 3-dehydrogenase that I described to you earlier. Thiamine deficiency leads to decreased ATP production. In our body, systems most sensitive to decreased ATP productions are cardiovascular system and nerve system. When symptoms are manifested in cardiovascular system, such as vasodilation, tachycardia, it is called wet beriberi. When symptoms are manifested in nervous system, such as difficulty in walking, speech, loss of tendon reflexes, and involuntary eye movement, it is called dry beriberi. When symptoms such as impaired memory and ataxia show up, it is called Wernicke-Korsakoff syndrome. All the three conditions are the results of thiamine deficiency, secondary to alcohol abuse. Now, let's look at the control of TCA cycle. For TCA cycle, there's no hormonal control. When I show this to my students, they are very happy because it means less work for them. But TCA cycle does have allosteric control. When I show this to my students, their minds start to rattle. Allosteric control, in some cases, can be quite confusing. But I told my students, for TCA cycle, allostatic control may be easier than you think. The key step for allostatic control in TCA cycle occurs in isocitrate dehydrogenase. And this enzyme is inhibited by ATP and NADH and activated by ADP. For example, during fat state, especially under resting conditions, metabolism of excess nutrients generate more energy than needed. What shall we do? We should slow down energy production. How can we do that? The first things we can do is to slow down TCA cycle, and that is achieved by inhibition of this enzyme. Inhibition of isocitrate dehydrogenase by ATP and NADH leads to buildup of isocitrate and citrate, especially when citrate level goes up. Citrate can be shuttled to cytoplasm. Rising level of citrate in the cytoplasm inhibit a key enzyme in glycolysis, and that is PFK1. When PFK1 is inhibited, glycolysis comes to a grinding halt in most tissues. So now, when the level of ATP and NADH goes up, we slow down TCA cycle and slow down glycolysis. So this happened in most tissues. But there's some exceptions. Liver is an exception. During fat states, liver can convert excess glucose to fat for storage. How does it work? Glucose is first converted to pyruvate through glycolysis, and pyruvate is shuttled to mitochondria, where it is converted to acetyl-CoA. Then utilize the first step in TCA cycle. Acetyl-CoA is converted to citrate, and citrate is then shuttled back to cytoplasm, and then converted to fatty acids. So in order for liver to convert excess glucose to fatty acids, these entire pathways need to remain open. How can liver overcome inhibition of PFK1 by citrate? So I asked my student this question. The answer comes from insulin. In fat states, insulin activates another enzyme, PFK2. PFK2 is the enzyme that synthesizes fructose 2,6-bisphosphate. And this compound is a potent activator of PFK1. Activation PFK1 by this compound overcomes inhibition by the citrate. TCA cycle also subject to control by availability of oxygen, ADP. When oxygen level is limited, electron transport chains is slowed down, and that will lead to buildup of NADH. What shall we do? We should slow down the production of NADH, and this can be achieved by slow down the TCA cycle. And this occurs by inhibition of isocitrate dehydrogenase by NADH. So these results indicate regulation of TCA cycle and electron transport chains are coordinated. On the other hand, when oxygen is abundant, TCA cycle is controlled by the level of ADP. When the level of ADP goes up, it signifies the need for energy. How can we meet the demands of ATP? So we can provide more NADH, FADH2, this high energy compound, to the electron transport chains, and so that they can produce more ATP. So how can we increase the production of this high energy compound? And the answer comes from activation of this enzyme. Activation of isocitrate dehydrogenase by ADP increase the production of high energy compound by TCA cycle. So this again indicates regulation of TCA cycle and electron transport chains are coordinated. This information is also described on these slides. These are the references that I used to prepare lectures on biochemistry, genetics, and molecular and cell biology. Thank you.